Hello, my name is Rubio and I'm going to present the poster Trees and Democracy in Precision Medicine. The authors of the poster are Katina Sada, Juliana Imperio, Enrique Montal, Paul Lasqueta, Maria Peña, and Itziar Abadia. So I'm going to divide this presentation into four main topics: the introduction, objectives, methodology, and results. Precision medicine consists of giving the proper medicine, the proper drug to each of the patients based on the different medical records and omics data that we have for each of the patients. Precision medicine also implies to decide which is the proper dosage for each of the drugs. Usually, the information that we are going to have for each of the patients consists of genomics data, maybe gene expression, or maybe mutational data, the data related with the lifestyle and the electronic health records. And we have the different drugs, and we want to find which is the proper drug for each of the patients. We are going to apply the precision medicine for the acute myeloid leukemia. This is a cancer of the blood and bone marrow, and there are a number of therapies, but the standard of care of AML has remained substantially unchanged for the last 40 years. Nevertheless, there are new treatments that are worth to be studied. So in this poster, our aim is to study the drug response in AML patients, and to do that by implementing several tree-based models to find which is the optimal drug, and to find and to tune the hyperparameters of the model to find which is the one that delivers the best performance. So the methodology that we are going to use, since we can't treat patients with several drugs, what we are going to use is to use a data set that is called the BEAT AML, and this data set consists of 257 ex vivo cell lines extracted from AML patients, and each of them have been treated with 111 different drugs. We have also information on different expression and mutational data from these patients. First of all, we can use this data set to find the oracle, so which is the best drug for each of the patients. The way to do that is to find which is the most effective drug based on the IC50 of each of the drugs on the cell lines. And we will compare this oracle with different tree-based methods. We are going to use three of them. The first one is the optimal decision trees, in which in each split of the tree we are deciding which is the variable, mutation, or expression that best splits the data, the patients, into two different groups, and we assign a drug for each of these two different groups, and this process is repeated iteratively until each of the groups is smaller than a preset hyperparameter that is the minimum number of patients for each group. We will also use a random forest and the extreme gradient boost methods. The question for these two last methods is that the extreme gradient or the random forest methods are not suited for the precision medicine problem, that is to find which is the best drug for each patient. So what we did is to establish a voting scheme based on the response of each of the cell lines to the different drugs. We are going to give a number of votes to each patient, and these votes of each patient will be given to the different drugs based on the response. We have several parameters to decide how to give these votes to the drugs. The first and most immediate one is to take the strategy winner takes all. So we can assign the best drug for each of the patients. But there are other possibilities. Using these different voting schemes, we got this result. In this case, what we see here is that including the voting scheme outperforms the simpler approach, the winner-takes-all method, and using 10 or 20 votes for each of the patients outperform any other method. That using expression slightly outperforms using mutations for the same method and the same number of votes, although the difference is small. On the other hand, the extreme gradient boost and the random forest clearly outperforms the ODT method, that is the yellow one in the figure compared with the bluish and the reddish methods in the figure. Extreme gradient boost is slightly better than the random forest, and the results for 20 votes are on the verge of statistical significance. So it's a little bit better, the HG boost. And on the other hand, ODT is much faster than XGB and random forest, has less hyperparameters, and is much more easier to it's much more easy to give an interpretation to it. And that's all. Thank you for your attention, and I hope to see you in the presentation in the conference.